lead discovery and identification is a major component or step in drug discovery. Once we have done with the lead discovery, we want to have lead optimization. So in lead discovery, we have multiple or many candidates, and now we want to filter out those candidates which should be moved towards preclinical testing phase. So how lead optimization is performed and what we understand from this lead optimization. So as the name suggests, lead optimization, it should be done to remove extra leads, that if there are multiple options, then we have just very selective options available, and then useless options should be removed so that time can be saved, because it is a very expensive process. So what is basically happening in lead optimization? We want to optimize our desired trait of the lead. So desired trait are part of the lead which we want to obtain. Then it should be based on chemical characteristics of the leads. So chemical characteristics means which are part of the inner side of different leads. And finally, methods from lead identification can be used for lead optimization also. So when we talk about lead identification methods, there were 7 different methods such as structure activity relationship, NMR based methods, and so on. So which methods can be used in lead optimization? We will just focus only on one method, which is basically our QSAR, that is quantitative structure analysis relationship method. What is the advantage of this method is that it focuses on the inner structures of given molecules to find out what is the inner structure, and then it tries to find out homologues, differences, and then find out which molecules will have higher potency and higher selective values. Similarly, it improves our physiochemical and drug-like properties. So physiochemical means which are combination of chemical plus physical sciences, and then which have more chances to become a potential drug. So once we are done with this lead optimization, successful drugs are moved towards preclinical test and then animal testing, and later on to humans, and later on to distribution phases. So what are different lead optimization methods? So we have very common de novo methods. So de novo method, basically they are our novel methods which are based on non-peptide molecules taken from ligands, and then these non-peptide molecules or ligands, they are able to bind with other molecules to make a successful drug. What are different properties? As we said, that they should have very good chemical properties such as lipophilicity. So lipophilicity basically is the property of a molecule to absorb in some chemical compounds other than water, such as in oil type things, oil and fat type things. Then we have pKa type things. pKa value, basically it tells the value of a substance to dissolve in acid type things. And then we have side chains and H bonds, donors and acceptors, so that they can have relationship with other chemical compounds with the help of their bonds, either in accepting or receiving some information. Then we have structure based drug design. So what is the effectiveness of this structure based drug design? Since it is a 3D structure, so it gives complete information of our inhibitor and what are different targeted drugs. Then we can have large number of medicines because we have complete information in the form of 3D structures. So very huge number of medicines can be generated. Then we have molecular recognition in protein ligand, and then in protein ligand complexes also. So what are protein ligands and what are their complexes? So if they are mono or multiple, then we can use this information to make our drugs. So what are drug-like properties which are used for lead optimization? Such as in phase 1 we have admit properties, which was our absorption, distribution, then metabolism, excretion, and non-toxicity, which was from phase 1. Then we have different filters which tries to find out similar information and useful information from our proteins and removing the extra information. Then we have bioavailability, PK, CNS values, which are taken from pharmacokinetics domain. So these are different parameters or properties of drug-like things. So once all this lead optimization is done, then our information goes towards preclinical testing or animal testing. In animal testing, we have a concept of xenograft models. So xenograft models are basically when we have a animal, and what we do, we implant some human genes into some animal and then see effects of some drug on that specific gene which is taken from human and put inside some animal. So those are xenograft animals. Again, based on this xenograft model, we want to see what are different admit effects on these methods, and based on these admit methods we perform validation and testing of our produced drugs.